Okay, so couple quick things just as we go through. So we're going to be in here, unfortunately, every day of the week, Monday, Tuesday, Wednesday, Thursday, from 9 to 11, well, 40, it should actually say. The other thing I want to quickly just say is that I'm going to have office hours every afternoon from 2 to 3:30. I should be in my office as well, except for late Fridays. Um, you can also feel free to email me then anytime you guys might need some help. What I really just want to say is while I got some office hours every day this week, you can also talk to me before class, after class. We're going to be going through quite a bit of content over the course of the semester or the winter term. And that is going to go pretty quick. So the instant you guys think you might need any help whatsoever, talk to me before class, after class, during some breaks we're going to take during class sometimes. Come my office hours in the afternoon. It is going to be an intensive course. With this immunology course, we are going to cover immunology and the same amount of content as if it was a full semester long class. Each particular day in class, each lecture day is the equivalent of 1 week in the regular semester. So it is going to be covering quite a bit as we go through it. With immunology, there is a recommended textbook. You don't have to get the textbook. It's the Kubi Immunology textbook, but that's the one that I'm largely going to stick to. I do have several things that are outside of the textbook at times. I do kind of use a few different textbooks, but the Kubi Immunology textbook is probably one of the better immunology textbooks. That being said, I'm actually still not particularly happy with it. There isn't actually a very good immunology textbook out there. They don't have a ton of figures and they're very technical in their language. And so what I really want to recommend is the textbook is good. The textbook has some extra stuff that we aren't going to go through class. But really what I'm going to be assessing you on is what we talk about in class. And so that is where you want to focus in is what is covered in the lectures, what's in the lecture notes, what we've talked about during class then as well. The other thing you do want is the Communication Statistics in the Sciences handbook, the orange cover handbook that has things like how to write papers and how to do some other stuff there. We are going to be writing some papers as part of this particular class this winter term as well. With immunology, we're really going to be trying to developing a detailed understanding of the mammalian immune system. And we're going to talk a lot about cells involved in the mammalian immune system. We'll talk about molecules, signaling molecules, and other things that control those cells that then mediate our immune responses. As we go through all of this, we are going to take a detailed look at how our bodies protect us from disease. But then obviously we still get sometimes sick. So then there's some limitations of the mammalian immune system then as well. And we're going to talk also about what happens when the immune system doesn't work properly, or when the immune system, even worse, starts to damage our bodies instead of protect it with things like autoimmunity then as well. So we're going to talk a little bit about this, but we'll talk about different parts of our immune system, like innate immunity versus the adaptive immune system. What are some of the components of those? How do they work? How do they protect us from against disease? But we'll talk also about things like hypersensitivities or allergies. We'll talk about autoimmunity. We'll talk about cancer in the immune system. And we'll talk about some immunotherapies then as well, and vaccination. Those are some of the main topics we're going to cover during this particular class. How are you going to get evaluated? There's a couple of different things. One, attendance is mandatory for this class. And in fact, I give you points towards attendance, 5 points per lecture day. So 5 points for Monday, Tuesday, Wednesday, Thursdays. The reason I give out points for attendance is because we're going to go through a lot of material on each particular day. And that means you should be here present, should be paying attention. We're going to do lectures plus some activities on Monday, Tuesday, Wednesdays, and Thursdays. So winter term this year is 3 weeks. It's this week, next week, and the third week. Okay, on Monday, Tuesday, Wednesdays, and Thursdays, we're going to be lecturing and doing some other activities like some in-class discussions and some other things. And I am giving you points for that one. There's going to be points for attendance. There will be also some discussion and participation points, especially during some more formal discussion class sections. On Thursdays, we're going to have a discussion-based class for about 45 minutes or so instead of a full lecture period. Okay. Those are going to be worth points. The reason is then those 4 days are then going to lead to an exam on Friday. So we are going to be lecturing today, Tuesday, Wednesday, Thursday. You guys have your first exam coming up this Friday. And that's the way this is going to work. We're going to have 4 days of lecturing and content, Friday exams. And Friday's just going to be exams and that's it. Okay, we won't be doing anything else those particular days. Questions about that so far? I do have content worksheets or quizzes on Brightspace. Your first one is due tomorrow. And I'll show you some of those here in a second. But we will have daily, it says daily, but there actually is, you're going to have one due on Tuesday, Wednesday, and Thursday is the way it's really going to work. So you got your first one due tomorrow. You have another one due on Wednesday. You have another one due on Thursday. The following week, again, we're going to follow that same pattern. Worksheets or quizzes that are on Brightspace, and they're due Tuesday, Wednesday, Thursdays. Okay, and I'll send out some reminders about that. Again, those are on Brightspace. You go onto Brightspace, you're going underneath the content tab of our Brightspace course sites. There is the syllabus loaded onto there. And so you guys should probably take a look through that if you haven't already. I do have lecture outlines posted onto there. So for instance, here's the first week's worth of lecture outlines, and you'll notice most of these are pretty long. Okay. I do have the worksheets or quizzes, and I'll get this in here shortly, but these are posted up onto Brightspace, and I'll get those populated into those after class then as well. And I'll send out an email reminder. I am going to have recordings of lectures. You guys probably noticed I started recording here. I do have recordings. It's recordings. Don't rewatch recordings. We're already going to be in class here from 9 to 11:40 each day. Watching a recording is not a good way to go through material because it just takes too long. It is a good way that if there's one particular topic you wanted to go back through, you can pull that part of the lecture up and go back through it if you need it. But that's why I'm going to do some recordings. I also record just in case anybody is out sick then as well, so they can keep up with the course content. All right, exams. Again, there's going to be 3 exams throughout winter term here. They're on each Friday of the class. So Friday, January 10th, Friday, January 17th, Friday, January 24th, which is the last day of winter term. Each exam is worth 100 points. Exams will cover any assigned readings, but as well as the material covered in our lecture classes. The exams, okay, so are predominantly going to be over lecture material, but there are some readings we're going to go through, and I'll cover those here in just a second. Exams will predominantly be over the new material. So for instance, exam number 1 will be over week 1 material. Exam 2 will be primarily over week 2 material. Exam 3 will be primarily over week 3 material. However, one of the things you guys are going to take note of is that a lot of the stuff that I talk about here in week 1 or week 2 is going to come back up later, and then we're going to add some more stuff to it. And so there is a little bit of a comprehensive portion to each and every exam. So exam 2 will have some exam 1 material back on it. We'll go through some of that exam 1 material again in exam 2, and it'll be back on there though. But there is some additive stuff. Immunology, the immune system is really complex, and there is a lot of this part talks with this part of our immune system, which then controls this other part of our immune system. There's a lot of crosstalk between the cells and the molecules involved in our immune system. And so those are going to be things that come back up at times. The other thing we're gonna be doing, is I have right now on Brightspace, readings for you for throughout winter term. And if you guys want, you can get an early start on all of these. But each of the 3 weeks, so I'm gonna have you guys read one primary research article and one secondary review article. These are already posted on Brightspace. So if you go and look in Brightspace, for instance, right now, I have the little section that is labeled material for summary papers and class discussions. If you go click on that one, it will have an assignment. This is a summary paper and discussion assignment description. So this is what I expect out of 2 kind of different parts, a summary paper that you guys are going to have to write each week, and then an in-class discussion we're going to have each week. For that one, then there are articles. I have a primary article for you to read. And if you read that and bring it up, it is kind of just a primary research article for you guys to read. I also then have a secondary review article. The one that I have for this first week is on NETs, neutrophil extracellular traps is what these are called. So we're going to talk about neutrophils a little bit today and tomorrow. So neutrophils are going to come up quite a bit in class today, in class tomorrow. This is going to be a paper over kind of neutrophil function, but also an interesting little thing they do that's part of our immune system. So I have a primary article, an older one from 2004. I have a more recent review article on NETs, neutrophil extracellular traps from 2022. So you're going to read both of these by Thursday at latest. Actually, you should read them by Wednesday at absolute latest. And then what you do is we're going to do 2 things. You have to write a 2-page summary paper over the primary article. So it's 2 pages, double spaced, 12-point font, Times New Roman. Okay, all this is described up in the assignment then. So if you pull up the assignment, it has a description of the assignment, but it has the 2-page summary. Again, it's supposed to be typed, double-spaced, 12-point font size in Times New Roman font is what I want from you guys in Word, Microsoft Word, or something similar. But basically what you're going to do is it's just 2 pages. Half a page should be basically describing the introduction or background of the primary article. 1 page is the results description, and then half page of conclusions. So 2 pages total, but you're focusing in on background material, on the results in the paper, and then on the conclusions in the paper. You're summarizing what the paper found. You do not have a separate section for the methods. Instead, the methods are described as part of then the results. You don't have to describe the methods or in essence, you just have to list what methods were done roughly when you're then also describing the results. And I have kind of an example statement here in the assignment description. When T cells were stimulated with interleukin-2, expression of gene X increased significantly as measured by acute RT-PCR. This is the method that was done. You're just saying what method was used, and then this is the result, in essence, that was observed. That gene X expression went up. Does that make sense, everybody? Okay. So summaries should be detailed, not simply a rehash of the paper's abstract. 2 pages is much longer than the abstract of the paper. So I'm asking you to go into more detail than the abstract. You should not be plagiarizing or copying the abstract for this particular assignment. I want you to not go over 2 pages, but I want you also to get simultaneously as close to the full 2 pages as possible. Does that make sense, everybody? This can be difficult to fit into 2 pages when done correctly because you have to be both detailed, but also concise. And I do expect you to describe every part of the paper's results. So you should be hitting all of the major results and figures from the paper and describing those then. Okay. Questions about that summary paper? Okay. These are due Thursdays. Summary papers are due Thursdays. The other thing, though, is we're going to have in-class discussions on Thursdays over those. And so this is where, and we're going to have the summary paper is over only the primary article. That's the other thing I forgot to say. The summary papers are only over the primary article. The discussions, we're going to have an in-class discussion. The discussions are going to be over both the primary article and also the secondary review article. Do you guys know what the difference is between a primary article and a secondary article? Primary articles are journal articles that go through experiments and that were written by the people that did the experiments. So a primary article, researchers that did the experiments wrote the article on that particular topic. So it's a primary or first-hand account of research. Researchers did the article, did the experiments, wrote the article over those experiments that they themselves did. Secondary articles are often commonly known as review articles. Secondary articles is when there is an expert in the field, and what they do is they summarize several different primary articles into one spot. They may not have done the research, but they are considered to be an expert in the field, and they summarizing what is the current things we know on this particular topic at the moment. And they make sure to cite where all the information came from all the different primary articles that they're summarizing. Some of the review articles that I'm giving you are actually citing like 150 other articles. Okay. And then they all go into one spot. But that's a review article. It's a current body of knowledge on a particular topic. Questions about that? Okay. The discussions on Thursdays, we're going to do this for probably about 45 minutes or so, but we are going to basically, you guys are going to, I expect everybody to participate. I expect everybody to be vocal during these. I'm going to make sure that I'm keeping track of who's talking and who's contributing during some of these. But as part of this assignment, I do have some things for you guys to kind of think about as you go through these articles. You can actually, some questions that you could think about to help facilitate discussions. I have a little table in here of questions you probably want to keep in mind when you're reading some of these articles that you might want to make some notes of and some other things. It also helps if you've done your summary paper before the class discussions, because then you've gone through the articles in detail, you've prepped your summary paper. You can bring your summary paper, you can bring copies of the articles to the discussion with you and use those to help you participate then as well. Questions about that? I also have a rubric, how I'm going to be grading your summary papers on this assignment description then as well. Okay, so make sure you're going through, again, it's materials for summary papers and class discussions. Right here at the top is the summary paper and discussion assignment description. Make sure you guys are going through that. Make sure you're taking a look at the question list to help help you prep for discussions. Make sure you're taking a look at the rubric on what my expectations are and how I'm going to grade the summary papers then. Questions about that? Okay. I'm doing this because we're going to cover quite a bit of, sorry, whoops. All right. The other thing is there is, oh, sorry. Yeah. Okay. The other thing is there is a more formal, larger paper presentation then as well. So this is separate. I'm asking is that each student selects a immunological topic from a list, or you can go off list with instructor approval. And I'll show you the assignment and the list here in a second, where you're going to research it and prepare a paper presentation. The topic, I want you guys to try to have a topic or a couple different topics that you're thinking about doing this paper on by this Friday, January 10th. I'm going to have you turn in your paper topics on Brightspace to me. Okay, so there's going to be a little worksheet quiz thing module on there, but you're just going to log in, and just for 2 points, you just have to tell me what topic you're thinking about doing. That doesn't necessarily lock you into the topic, but I want you guys to be thinking about what you're doing for this one. You're then going to do a paper presentation. The paper presentation is going to be similar to a secondary review article. And this is part of the reason why I have you reading secondary review articles, because then you can see how those are organized, how what goes into those types of papers, and then you're going to be mimicking that format for the final paper that you have to. This is due Friday, January 24th. That comes up fast. What that means is don't wait till the last week to start all of this. Go ahead and get it started and out of the way now, okay, as early as possible. Start working a little bit on this every particular day. And this final paper is worth 50 points of your grade. With WAS, there is on Brightspace then a paper presentation tab. And if you go into there, there's a drop box for turning in your paper, but there is then the paper presentation description. And so on here, it has, again, what the description of the project is, what's the due date, paper expectations. I am expecting you guys to have a minimum of 6 primary and or secondary sources cited in this paper presentation. This is not things like the CDC's website. That's a fine website to use, but it does not count towards the required sources. Okay, they need to be primary journal articles or secondary journal articles. I've got a description of databases you can use. You can use Google Scholar at scholar.google.com. You can use PubMed. You can use EBSCOhost on Brightspace and the library Brightspace page here at Mount Mercy. But you're going to use one of these professional databases to then find professional sources. You need at least 6. That's quite a few. Okay, so you want to get an early start picking a topic. You want to get an early start researching that topic. With some of these topics, some of these topics are a little easier to do background research on them than others. Some of the topics, you got to have the right keywords when you're doing some of these database searches. You have to know how to use the database to narrow down to the right references that you're looking for. If you're having trouble finding good references, just come talk to me. I'll show you how to use some of these things, and we'll also try to find some keywords that is going to work better for you to narrow down the topics to what you're looking for. The other thing is when you're looking at references, I recommend something called citation chaining. Try to find some review articles, some recent review articles first. But if you really want to find more good references, just track what they cited backwards. Citation chaining is where you, in essence, you find a paper that's really good. You want to find more papers? Well, what you do is you just look at what they cited. And you're going to usually find some more good papers from that then as well. You can actually do citation chaining backwards. This is where you find a good resource and you look at what they cited and go backwards in time. But you can also sometimes do citation chaining forwards. Some of the databases like scholar.google, Google Scholar will let you see, I found this really good paper, but it's kind of old. What paper cited it? And so you can actually see this particular paper was cited and used by these other papers more recently. Does that make sense, everybody? So this is called citation chaining. Citation chaining really speeds up finding good literature citations, good references. Questions about that? So you're going to make a paper. I have a list of suggested topics. I've given you quite a few topics. It's over a full page of a list of different topics. But you could do things like, what is the role of toll receptors in the immune system? How are cytokines regulated during pregnancy? How is some different aspects of cancer immunotherapy, which is using the immune system to treat cancer. You can have things like vaccination, development process of creating and mass producing vaccines. Vaccination as a treatment for cancer. And yes, there are some types of vaccines they're looking at potentially as cancer treatment then as well. Some of these are still in the developmental stage or the testing stage. So there's some answering. CAR T cells, COVID, immune evasion by SARS-CoV-2, SARS-CoV-2 and cytokine storm disorders. There's a number of different topics. Take your time, look through the different topics and see if there are a few that catch your interest. And then that is something that we can look through. You guys are also free to come meet with me and I'll give you some background on some more details, some more background on some of these topics then as well. Okay. Again, I have a grading rubric here at the end that shows exactly what I'm looking for. And these then paper assignments kind of breaks down what would be kind of a excellent, good, or acceptable version of some of these then. Okay. Again, do remember though, at least 6 primary and or secondary references minimum for this paper then. Questions about that? Again, there is, the earlier you get started on some of this stuff and get it out of the way, the better, typically. The longer you wait, the more stressful winter term gets, which is not my goal from all of this. My goal from all of this is that the human immune system is very complicated. We're going to be covering, still detailed, but we're just covering the basics of the immune system. The reason I have you reading some primary and secondary research articles is just so that you guys can see what goes into some of these, but also start to explore a few different topics in a lot more detail then. So it basically is a way of just covering some topics in class in more depth. It shows you how complex the immune system is and exposes you to material that we would not have time to cover in more of a lecture-based content. But I just want you guys to be aware of how complex and complicated some of the stuff that is being done. And then a lot of times these textbooks are really taking decades and decades of research from so many different research groups and condensing it down. And I kind of just want you guys to get exposure to papers that contributed to building up all that body of knowledge. Okay. The other thing is, again, we don't have time to cover every aspect of the immune system. And so the big paper presentation, the 50.1 that's due at the end of winter term, that's also basically my way of letting you guys choose a topic that you are interested in and then learning more about it in case we don't have time to fully cover that in class. All right. Grading scale, if you need this for the major, for anything, you need at least a C. Brightspace, I already showed you quite a few things, but grades will be accessible real-time through Brightspace. Outlines of lectures are uploaded those. Lecture recordings will be on to there. But we have announcements, copies of the quizzes and worksheets, the paper presentation drop boxes, the discussion material, and all the other stuff will be through Brightspace then as well. Okay. How to succeed. Do use the outlines to take notes more effectively. I will be going through things pretty quickly. We will cover a lot of material each particular day in class. Come to lecture prepared ready to go. Participate. And that also means not just participate in discussions, but make sure that you're asking questions or anytime you guys think you might need a clarification. If you're not sure about something, talk with me before, after class, or in office hours. Start again, I've said this already a couple times, but I'm going to emphasize it. Start any paper presentations immediately. Time goes by very quickly. We're already going to be in here for 2 hours and 40 minutes every morning. And so you need to be doing all of this material and getting it done and out of the way quickly, or it gets more stressful than it should be. Set aside time to study a little bit every day still. Again, winter term goes fast, and we're going to be lecturing Monday, Tuesday, Wednesday, Thursday. And then we have the exams right away on Friday. By the way, I've taught immunology both in winter term and in a spring semester previously. Actually, the class usually did better in the winter term format, strangely enough. I think it's because we're going to go through a lot of content, but then I'm going to immediately give you an exam on it. And what ends up happening is, yes, you need to study and prep for it. Yes, you need to be going through that. But it also means is a lot of times the material is a little fresher in your head by the time the exam comes around. Does that make sense, everybody? Okay. All right, ask question, make suggestions. I always want you guys to be vocal during class. I want you to stop me if you guys have any questions or need anything, or if you just want to hear about like something related to what we're talking about in class. It's always more interesting when you guys get to hear what you want to hear, okay, and what you want to hear about. Okay. Questions about any of that? So we're going to be talking about a few different things here, but again, we're going to be focusing mostly on the immune system. Now, in immunology, this is the study of immunity and the immune system. Immunity or munus is Latin for exempt. Immunity then is where we exempt diseases from our body. It's a state of protection from disease. Immune system then is going to be a series of bodily defense mechanisms that is then going to be mediated by a number of different specialized cells that are often collectively referred to as white blood cells. So we're going to talk a lot about different kinds of white blood cells. So its immune system has a number of different interacting specialized cells, white blood cells, and then molecules that those white blood cells often make that are designed to recognize, respond, and destroy any foreign things to our body. Could be a virus, could be a bacteria. Sometimes, unfortunately, it's things like pollen, if anybody has hay fever, though, for instance, which pollen is harmless to us, but our immune system still recognizes it as foreign and tries to fight it off, even though it doesn't really need to. Now, the concept of immunity has been known for a long time. And if we go back into kind of historical writings, again, there's even things back into the 400 BC by Greek historians. They're describing things like people who got sick during like disease outbreaks. They were being used as nurses to help treat, for instance, people who got sick because they noticed that they could not get sick from the disease a second time. So people who got sick from a disease, recovered, they were known to be immune against that disease. And they were often taken in care, in the case of disease outbreaks, they're often used as caretakers for the sick individuals. Okay. So this has been, the concept of immunity has been known for a long period of time. This is, you guys probably heard of smallpox before. Smallpox is very, very bad. Smallpox, first off, causes, this is the kind of the blistery rash that it causes. This can be disfiguring for life. So a lot of times those nodules, they scar over afterwards. The other thing with smallpox, and one of the reasons it's particularly bad and why a lot of researchers worry about it, is it's fatal 30% of the time. So 30% of people who get smallpox die from it, in essence. And so this one was actually a big problem And they started doing things like variolation to protect against smallpox, even back into the 15th century. Variolation is basically a rudimentary version of vaccination. What they would do is they would have somebody with smallpox, but they were starting to recover. They were getting noticeably better. What would end up happening is they would poke needles into the blister nodules where the smallpox virus was. And then they would take this and they would introduce very low doses into people on purpose. So they would poke the needles, get some of the pus, and then they would poke other people with it to get a thing. But they always did this in recovering patients, patients that were starting to get better. Because the patients were starting to recover and get better, the virus was still there, but it was often weakened. The immune system was starting to destroy the virus in those recovering patients. They would then intentionally just basically poke a needle into that sick recovering person, but then poke that needle into somebody that's normal healthy. It was a very rudimentary version of a vaccine. In many cases, the person might then get a minor case, but then would recover and develop immunity against smallpox. The problem with this was it doesn't always work, and sometimes it literally gave the person smallpox, and then they got sick with like full disease from it instead. So it wasn't a very good version of vaccine, but they did this because it was still better than getting the disease itself, typically. And this was done all the way back in the 15th century in China and Turkey. Later on in the 1700s, Edward Jenner actually then developed the first kind of more vaccine. What he noticed is that milkmaids that were milking cows, sometimes these milkmaids caught a virus called cowpox. Cowpox is very similar to smallpox, but it's not the same virus. And cowpox infections are typically not fatal and not disfiguring. The person just got a little bit sick, but then they recovered. But it actually, cowpox virus is so similar to smallpox virus that anyone who had cowpox, he noticed, especially like milkmaids, they were immune to smallpox and they didn't get it. So this was then they started giving people cowpox to then prevent smallpox, in essence. And so this was, again, one of the first cases where we're using the immune system to try to treat disease, in essence. This then he termed, Louis Pasteur came along and he actually coined the term vaccines. Vacca is Latin for cow. So the name vaccine basically is from the Latin word for cow, but it's because kind of what is considered the first modern day version of vaccination was using cowpox to treat and prevent smallpox, in essence. Again, they could take attenuated cultures of bacteria from chickens or fowl and use those as a cholera vaccine. And he also extended this to basically then doing a anthrax vaccine. So Pasteur turned this vaccine, he named it after Edward Jenner's work, but he actually then started making in the 1800s various different vaccines to, again, things like cholera, to anthrax, and then Joseph Meister actually took also some of his work and developed the first rabies vaccine then as well. Immunology has been around for a while, but this is a big area of research. This is actually, I actually need to update this a little bit here, but this is a listing of Nobel Prizes that were given out for research just into immunology over the years. There's a lot of people that do research in immunology. This is people that have won the Nobel Prize for some of the research. And in fact, one of the papers that I'm having you read next week is from a researcher that won a Nobel Prize. And it was the kind of research that started a whole field of immunotherapy then as well. So I'm going to have you read a paper next week that is in essence a Nobel Prize winning. They don't win a Nobel Prize for one single paper. They usually it's a bunch of papers on a particular topic that somebody wins a Nobel Prize over. Does that kind of make sense? But this is the paper that started kind of the entire thing. Yeah. Immunology. The flip side of immunology is disease. So we're going to talk a lot about immunology this winter term. We're also going to talk a lot about disease this winter term. Disease, if I'm going to define what a disease is abnormal conditions that affect an organism, causing noticeable symptoms and signs. So a disease is any time our bodies have abnormal conditions or are not working properly. And then you get symptoms and signs. Now, I do have a difference here between symptoms and signs. A symptom versus a sign. A symptom is some type of subjective change in the bodily function of the patient. The patient knows they have that change, but it's not readily apparent to an outside observer. There might be some examples of a symptom then. Fever is actually not a symptom. A fever is going to be a sign. Can you measure a fever? Can you feel a fever? Yeah, my kids, every time my kids are sick, they get tired of me constantly feeling their forehead. Anyways, yeah. Fatigue. So yeah, fatigue is usually considered a symptom. It's something the patient knows they have. They know they're tired and they don't feel well. That's not always readily apparent to someone else or easily measured. What might be another example of a symptom? Yeah. Pain is another big one. Pain is probably the best example of a symptom. Anybody ever been in the hospital or visited somebody? The nurses are constantly asking a patient to rate their pain on a scale of 1 to 10. You guys, anybody ever seen like that rating scale on the board in the rooms? It's because they can't measure it. So they have to ask the patient, what's their pain level today? And then they rate it on a scale of 1 to 10, for instance. So malaise, that's fatigue. So malaise is fatigue or tiredness. Pain. Another example of a symptom would be nausea, for instance, then. So those might be some examples of symptoms. Signs then are going to be objective changes in a patient that can be observed or measured. So these are observable, measurable in some way. Aiden, what's an example of a sign? Yeah, okay. So fever might be one example of a sign. What might be some other examples of signs? So inflammation is typically you got swelling, redness. It's a lot of times an inflamed thing feels warmer to the touch. So yeah, inflammation's a common sign. But something if somebody's sick, you can see them do. Or if you see somebody sick and they do something in particular, you might want to avoid that person if you're just sneezing, coughing. Yeah. Anybody ever had somebody cough in your face? Have kids, you'll get used to it. All right. Some signs are going to things like swelling and inflammation, fever, coughing, sneezing, could be lesions or rashes is going to be an observable thing. Could be vomiting, though, for instance, something that's observable easily. Now, with diseases, we're going to talk about several different kinds of diseases as we go through the semester. And sometimes what I'm just going to do is we're going to be talking about a particular topic, and then I'll just going to stop and we'll talk about a disease that relates to that particular topic. Sometimes we'll switch gears a little bit here. Different diseases, though, have different causes. Some diseases are example of what are called internal dysfunctions. This is where the disease is caused by some inherent or intrinsic part of the body going bad in some way. So it's part of the body just in and of itself stops functioning properly. These are called internal dysfunctions. So the inherent part of the body and intrinsic part of the body is defective in some way, and this causes abnormal conditions. What do you think I'm talking about when I'm talking about internal dysfunctions, or what might be some examples of diseases caused by internal dysfunctions then? So autoimmune disorders, the immune system starts damaging the body instead of protecting the body. So an example of an internal dysfunction is autoimmune disorders then. But there's several other kinds. Can anybody think of another example of an internal dysfunction? Cancer, yep. Cancer would be another example of an internal dysfunction. Okay, and I'm going to go through some more examples here in a second. Some diseases, though, are external dysfunctions. External dysfunctions is where something from outside the body gets into the body and then mediates disease. So external dysfunctions are caused by some type of outside disease agent. And when I say an outside disease agent, what do you guys think I'm talking about? What are some examples? Flu. So flu would be an external dysfunction because the flu is caused by influenza viruses. Viruses from outside our body get in and then they cause disease. There might be some different other examples of external dysfunctions. Flu might be an example of an external dysfunction. Can you guys think of any others? Huh? Allergies are actually going to be an internal dysfunction because that is actually caused by our immune system overreacting to a harmless thing, is typically what that is. So those are actually considered internal dysfunctions.
Bronchitis.
Most cases of bronchitis are brought on by some type of virus or bacteria. Yeah. COVID might be an example of an external dysfunction. HIV, human immunodeficiency virus, gets into our body and then causes AIDS, acquired immunodeficiency syndrome, for instance. So typically when we're talking about external dysfunctions, we're talking about bacteria or viruses or fungi get into our body, for instance. All right, again, some more specific examples. Internal dysfunctions. Genetic diseases, like, for instance, sickle cell anemia or cystic fibrosis. Those are going to be genetic disorders, genetic diseases. Those are going to be internal dysfunctions. You inherit those from your parents. Cancer is an internal dysfunction. Both autoimmunity, where the immune system damages the body, but also hypersensitivity or allergies are classed as internal dysfunctions. Some kinds of immunodeficiencies are internal dysfunctions. A lot of times, though, also environmental factors, yes, like pollen that causes allergies, are lumped into this category because the pollen in and of itself is not damaging. It's our immune system reacting to it that causes the damage.
Would you apply that to like carcinogens and stuff like that too?
Can be sometimes, yes. So for instance, smoking, you guys are all aware that smoking leads to a higher risk of what? Lung cancer. And so smoking is more of an environmental factor that leads to the cancer, but it turns out what you have to do is smoke for a long period of time. When you smoke, that leads to the introduction of compounds into your lung that is difficult for our immune system to fight off, but they still try to fight it off. And then the immune system causes chronic inflammation in the lungs. Chronic inflammation leads to damage over time, which leads to mutations accumulating in cells, which then leads to cancer. Hence, the environmental factor is lumped into because there's an internal component to it. Does that make sense? By the way, chronic inflammation of any kind can lead to cancer. That's something we're going to talk about later on here in winter term as well. External dysfunctions. are typically then some type of thing like a virus or a bacteria, a fungi, or a parasite like a roundworm or tapeworm. These get from outside of the body, they get into our body, they cause damage to the body, and then that will cause external dysfunctions, externally caused diseases. So external dysfunctions are typically caused by some type of microorganism, virus, bacteria, little tiny worm animals, fungi getting it. With that being said, are all microorganisms harmful? No, some microorganisms are harmful, but some are actually good for us. When we look at host microbe interactions, humans can interact with microbes in different types of what are called symbiotic relationships. And a symbiotic relationship is a close, intimate interaction. It's also typically a long-lasting interaction between two organisms. So with these symbiotic relationships, it's intimate. Usually means one organism is living on or inside of the other organism. That's what I mean by intimate interaction. It's usually a more long-lasting interaction than as well. So for instance, would a mosquito biting somebody be a symbiotic relationship? No, because they bite and then they fly off. It's not a long-lasting interaction. Does that make sense, everybody? Okay. There are three main kinds of symbiotic relationships, subtypes: mutualisms, commensalisms, parasitisms. Mutualism, this is where both partners in the symbiotic relationship benefit from that symbiotic relationship. Commensalism, this is where one partner benefits and one partner is unharmed or neutral. And then parasitism, this is where one partner benefits, it's usually the microorganism, and then one partner is harmed. Mutualisms are sometimes referred to also as a plus-plus relationship. Again, both positively benefit from the relationship. Commensalism sometimes is referred to as a +0 relationship. One partner benefits and one is neutral or unharmed in that. And then parasitisms are often commonly known as a plus-minus relationship. Again, because one partner benefits and one gets negatively harmed then in that relationship. With these, there's different kinds of examples. So for instance, we have about the same number of bacteria in our digestive tract as our own cells in our body. So there's different estimates on this, but it's actually estimated that an adult human is made up of about somewhere between 10 to 30 trillion cells. So you have about 10 to 30 trillion cells in your body. We also have about 10 trillion bacteria just in our gut, in our gastrointestinal tract. So you have about the same number of bacteria in and on your body as your own cells in your body. About 25% of the energy we eat is consumed by the bacteria. The bacteria living in our gut, they get a nice, warm, nutrient-rich, protected environment to live. So they're nice and happy living in our digestive tract. And they eat some of the food that we eat. Actually, it's estimated about 25% of your body weight is just bacteria. However, those microbes that are living in our gut also make things like vitamin. So for instance, Escherichia coli living in our gut makes vitamin K and vitamin B for us. We have no way to make vitamin K. Human body has no enzymes to synthesize vitamin K. We get all of our vitamin K from either it's already present in the foods that we eat or the bacteria living inside of it make it and share it with us. Anybody know why we need vitamin K? A couple different reasons. One of the main ones, though, blood clotting. If you have a vitamin K deficiency, your blood doesn't clot properly. And so, for instance, people with hemophilia, where they get cut and their blood doesn't clot, it's because they can't process vitamin K and use it. Another example of mutualism is something called probiotics. We'll talk about this a couple times. But probiotics, this is where we have live microbial cultures are either applied onto us or ingested more commonly. And that those probiotic cultures are then intended to exert a beneficial effect. For instance, probably too much information, but I had yogurt for breakfast this morning. Yogurt is full of bacteria. The way they make yogurt is they mix just milk and bacteria, and then the bacteria ferment the milk. And what you get is, everybody ever notice that yogurt's nice and kind of acidic, tangy? It's actually acids left over from the bacterial digestive process. But it takes that milk and convert it, then they add some flavoring and some sugar to it, and it's tasty. But it's full of bacteria, and a lot of those bacteria are good for our digestive tract. Those bacteria in our body, they do something called microbial antagonism, or well, sometimes what's also a type of competitive exclusion. Existing normal microflora that is good for us can prevent disease-causing microbes from growing up in our body. For instance, lactobacilli is a common bacteria in yogurt and milk. This is good for us. What it does is it is there first. And say you get exposed to Salmonella enterica. This is bad. This causes salmonellosis. Okay, food poisoning. But it turns out lactobacilli, if it's there first, it takes all the nutrients away from the salmonella and keeps it from growing up in our body. Because it was there first, it outcompetes the salmonella. We ever ingest a salmonella, then it can't grow up in our body due to competition with this guy. And then we don't get sick. It also turns out, though, that lactobacilli make some compounds that actively kill salmonella as well. So not only do they outcompete it for nutrients, they actually will actively kill it then too. It's trying to kill its competitors. So this is called competitive exclusion or microbial antagonism. These bacteria don't make us sick, and they can help protect us from bad bacteria. Questions about that? All right, commensalism. Many of the microflora associated with this are commensal. For instance, we have large population of bacteria that are living on our skin. But the outer layers of our skin are actually dead. Some of the, and we are constantly sloughing off skin cells out into the environment. And sloughing is the technical term for that. But we're often shedding skin cells continually. So there's bacteria that are living on our skin, they're eating our dead skin cells, but we're going to lose those anyways. So it doesn't cause us any harm whatsoever. Okay, bacteria get some food source, doesn't impact us in the end. By the way, it's actually estimated that 80% of dust in a home is actually dead skin cells. Just kind of creep you out a little bit. All right. All right, finally, we have parasitism, and this is where we're talking about pathogens. This is where a pathogen gets inside our body, infects our body, and then starts damaging and killing our cells. The pathogen is damaging and killing our cells as a way of reproducing or getting nutrients. So the pathogen is growing up, is happy, well, we get harmed in the process. And some examples of this might be strep throat, which is caused by the bacterium Streptococcus pyogenes. Or it could be something like Lyme disease, which is caused by the bacteria Borrelia burgdorferi, for instance, then as well. So those might be parasitic bacteria that cause disease if they get into us. With that being said, less than 1% of known bacterial species cause disease. So the vast majority of bacteria are neutral to us or sometimes good for us. There is very few bacteria that are known to cause disease. However, with that being said, about 80% of known human disease is caused by bacteria. Again, those are kind of difficult numbers to kind of tease apart. But the vast majority of bacterial species don't cause disease, but there's just so many bacterial species out there. Millions and millions and millions and millions of different species out there. And a good number of human disease is still caused by those few bacteria. The other thing that makes things a little bit more complicated sometimes is that relationships can change. So you can have a mutualistic bacteria can sometimes go and get transformed into a parasitic bacteria. So for instance, there's E. coli K12 versus E. coli O157H7. E. coli K12 is good for us. It's mutualistic. You guys all have it in your digestive tract right now, and it actually keeps you healthy and makes some vitamins for you. So E. coli K12 is a good version of E. coli. But there's also a version of E. coli called O157H7, and this causes food poisoning. And the difference between these two is about 10 genes. There's about 10 genes and gene products that can make a good bacteria into a bad bacteria. And bacteria like to do what is called genetic transfer. Genetic transfer is where a few genes will get transferred from one cell to a different cell. So sometimes bacteria pick up new genes and that changes them, changes their behavior. And sometimes if they pick up the wrong set of genes, a good bacteria can get transformed into a bad bacteria. And this happens all the time. For instance, Vibrio cholerae, this causes cholera. You guys get exposed to this all the time out in the environment. It's a normal environmental bacteria, but it's usually in a harmless version. If you happen to get exposed to the wrong version of Vibrio cholera, then you get, well, cholera, which is a diarrheal dysentery disease. Bordetella pertussis causes whooping cough. Again, this is a normal bacteria out in the environment. You get exposed to it all the time, but usually we get exposed to the harmless version of it. But sometimes you get a version of Bordetella pertussis that has picked up the wrong set of genes, and now it causes whooping cough disease. And for instance, in 2012, there was an outbreak of whooping cough here in Iowa. Which sounds like it was pretty far away, but that was the year my son was born, so I was really, really paranoid that particular summer. Anyways, because whooping cough is very, very bad for infants. All right, we're going to start off this semester talking about, instead of immunology, we're going to start off talking more about disease. But I've been talking at you guys a little bit long enough so far, so why don't we take a break? That usually means get up, walk around, stretch. Don't sit there in the seat, but do something to just kind of get the blood flowing and wake up a little bit. Let's take about a 5-minute break, and then we'll come back here a little bit later.